for a conversation on the microbiome. Please welcome Carolyn Edelstein, the co-founder and executive director of OpenBiome, and Jennifer Wargo, an associate professor at the MD Anderson Cancer Center. Here to lead the conversation, welcome Atlantic staff writer Ed Yong.
Thank you everyone for joining us. I'm very excited about this session because it touches on a topic that's very near and dear to my heart and my gut and other organs. I'm sure many of you will be aware that people and basically every other creature are full of bacteria and other microbes. They live in our gut, on our skin, and in our various other body parts. There are at least as many of them as there are human cells in our body. And it has become abundantly clear over the last decade of research that those microbes play an incredibly important role in our lives. They help to digest our food, they train our immune system, they help to build our bodies as we grow up. And it now seems that they have an important role in cancer too. There were some seminal studies several years ago showing that people's response to different cancer drugs depended on the composition of their microbiome, on the members of the communities that live inside their bodies. And that now seems to apply to some of the most interesting cancer treatments around, the immunotherapies that you've already heard about today. So Jennifer, you have done work on this. I would love to know how you got started, like what brought you into this field?
Yes, no, great question, and again, it's an honor to be here. So I'm a cancer surgeon. I started my career at Harvard University, and when I was at Harvard, I was treating patients with melanoma and also with pancreatic cancer, and we were interested in understanding why some patients responded and why others didn't. And we looked at the tumors of those patients, and interestingly, what we found is that in about 3/4 of patients with cancer, we actually identified bacteria within those tumors that could actually break down chemotherapy like gemcitabine. And so that's how I got interested in the microbiome, and so I really started looking at that. Later, a couple of years later, I was recruited to MD Anderson Cancer Center, wonderful place, and really great place to work with patients and with others to study cancer. And at the time, I had gone to a meeting where I saw the elegant work of Laurent Zipfogel and Tom Gajewski presented, who really pioneered the work in cancer immunotherapy with regard to the microbiome. And they had shown that in mice, depending on what the microbiome looked like within the gut of those mice, dictated whether or not they responded to immunotherapy. And so I was floored by the data and really wanted to know, well, I got up to the microphone after this talk and I asked, I was like, well, have you studied this in patients yet? Mm-hmm. And they hadn't at the time, and so I saw this as a really unique opportunity. So ran home to MD Anderson, wrote a protocol, enlisted the help of our patients, and together we, over 1.5 or 2 years, we collected samples from over 200 patients with cancer who were going on to immunotherapy and profiled the gut microbiome of those patients. And lo and behold, what we found is that if patients had a higher diversity of bacteria within their gut microbiome, they were more likely to respond. In addition to that, if they had different bacteria, so if they had certain bacteria or a more favorable microbiome, we also found that they were more likely to respond. And so we also took that and actually got samples from our patients and put that into germ-free mice, recapitulated the phenotype, really suggesting that what's happening, or what we think is happening, is that the microbes within the gut of those patients is shaping their immune system and how they respond to cancer therapy.
And what is the difference that you see between like the group that responds and that doesn't respond?
Right, and so we saw a couple of things. One, we saw more diverse microbes within the responders. We also saw that the responders had a higher abundance of certain bacterial taxa like Clostridioides, Ruminococcus fecali bacteria. And this was associated with more immune cells within the tumors of those patients. And interestingly, now we've gone back and looked at those patients with regard to diet and lifestyle factors. We actually found that those patients had a higher fiber diet. And if they actually ate more fiber and had this more favorable signature, they were 9 times as likely to respond to the treatment, which was pretty fascinating. And so now we're working with the Parker Institute for Cancer Immunotherapy and a company, Series Therapeutics, to run a clinical trial to actually see if we can change the microbiome and help patients respond better to immunotherapy.
Because this is one of the big mysteries of immunotherapy, that it seems to be kind of miraculous for some people, and yet others either suffer side effects or it just doesn't seem to work. And what your experiment suggests is that the microbiome is at least one of several factors that might influence that difference between the responders and non-responders.
Absolutely.
Okay, so Carolyn, your company is very much into this, involved in this idea of manipulating the microbiome, which is quite a challenging prospect. Can you tell us more about the work that you did and how you became involved in this field?
Yeah, absolutely. And I think our story is a lot simpler in some ways than the story of trying to intervene in cancer, but. In 2011, my cousin was living in New York, got sick, went into the hospital for a routine surgery, and came out with an infection called C. diff. And C. diff is a pretty aggressive gut infection, and oftentimes, actually, there are probably a couple of people in this room who are carrying C. diff, but usually the bacteria that are present in our microbiome and our guts tend to keep that C. diff in control, and it doesn't make you sick. What happened was, my cousin took antibiotics. It wiped out the community of microbes that was living in his gut, and from there, C. diff could spread and start producing the toxin that gives pretty aggressive diarrhea. So the solution was more antibiotics. He took them, but his infection kept coming back. And over the next year and a half, he was on 7 rounds of vancomycin, which is a last-line antibiotic. And after a while, he came across some of the literature on a procedure called a fecal transplant, which it's exactly what it sounds like. So there's very few euphemisms in this space, but you take a sample of stool from a donor, and in that stool are a lot of the microbes that you find in the gut. And by infusing the stool into the patient's colon, the bacteria from that healthy person's stool can outcompete the C. diff, and the infection is eliminated within a day. And so my cousin thought this sounded like a pretty good option for him, but he found one clinician in New York doing it at the time, and that clinician's wait list was about 6 months long. So instead, he took matters, I guess, quite literally into his own hands. He went home, got an at-home enema kit, asked for a sample from his roommate, and did the procedure himself. And it worked, but we thought that couldn't be how we were solving this problem. On the one hand, C. diff is the most common hospital-acquired infection in the country. About 500,000 people get it every year. But on the other, there was an at-home enema kit, and those seem to be the only things bringing together this solution that, at least in clinical studies, was showing that it was about 80 or 90% effective for these patients who just weren't responding to antibiotics. So it turns out frozen and fresh stool work equally well. So we started a stool bank, like a blood bank, but to make it easier for patients like my cousin to get access to the therapy.
So you provide stool samples from healthy donors for fecal transplants to sick patients with C. diff.
Yes.
My understanding of fecal transplants is that still, while there have been tremendous success rates for C. diff, it has been much less, the results have been much less consistent from trials of other diseases. So that even in this situation, when you're replacing you're transporting an entire microbiome into someone, it's quite difficult to really change a person's inner community.
Right, exactly. And so I think in C. diff, in some ways, this is where I was saying it's maybe a little bit more of a straightforward story, because you've got this opportunistic pathogen and you just need to push them aside to be able to reconstitute the microbiome in some ways. But I think in other areas where there's really active research, especially in inflammatory bowel diseases and in autoimmune conditions, it's a lot harder and a lot more complicated to achieve the sort of benefits that you may be hoping for, and also to sustain those benefits.
And Jennifer, do you see those sorts of complications in your work too? Is it going to be tricky to marshal the developments that you found, the links between the microbiome and the immune system and cancer, to actually affect the course of people's treatments?
Yeah, definitely. I think it's complicated, but I think we'll absolutely get there. The way to get there is to work together to do it. But, as Carolyn pointed out, I think in patients who have C. diff colitis, they essentially have a completely abnormal gut microbiome, and so they have one dominating organism. So if you actually can transplant a whole new microbiome, you can actually cure those patients pretty effectively. Now, in patients with inflammatory bowel disease and cancer, there's varying degrees of what we call a dysbiosis. And so simply transferring in a brand new microbiome from a healthy donor may not be the best strategy. And furthermore, the donors themselves, I think we need to better understand what's the optimal microbiome to transplant in. And, for C. diff and for other forms of colitis, it may be that any healthy donor is fine, but for cancer, we're not so sure. So we are actually within our trial using fecal transplant as well from our complete responder donors. So these are patients who had metastatic melanoma, got immunotherapy, and their tumors all went away for a long period of time. We profiled their microbiome, and they have what I would call a rock star microbiome. So those patients are actually, they're signing up to help other people and to donate their fecal samples in order to help potentially cure other patients with cancer. So, but there are a lot of complexities, and so certainly there's fecal transplant is one strategy you can use. You can also administer various consortia of bacteria, but again, I think we still don't know what's the secret sauce, if you will, and what's that magic mixture of 10 or 20 or 100 bacteria that will then, cure patients with cancer. We're not sure yet, but again, I think if we work together, we'll eventually get there.
And I wonder if it's clear what exactly the mechanism through that this is working through. Like, how exactly are the bacteria in the responders? What are they doing to give them to influence the way they're reacting to these drugs?
Right, and if you think about the gut, and you think about a very long tube with a very vast surface area, that's all along that surface area there are these microbes, 100 trillion or so, and just on the other side of those, where those microbes are, there's immune cells, and so there's a direct interaction between the actual bacteria and other microbes and these immune cells in our bodies. And so are these immune cells getting educated within the gut? That's certainly one possibility and known to be influencing it in some regard. In addition to that, if you think about the things that these gut microbes are producing, you think these vast numbers of microbes within our gut are actually producing things like metabolites that are then getting into our bloodstream and potentially influencing our immunity and other factors.
So I've got one more question for you two, but I want to then throw it out to the floor. So please get your questions ready. So I wanted to ask it seems like maybe we would need to personalize these kinds of treatments. Do you think that there would be a kind of one-size-fits-all rockstar microbiome that would suddenly make everyone super responsive to immunotherapy? Or would you need to tailor the cocktail of microbes that a patient receives?
Carolyn, do you want to answer first?
Sure.
As well,
Yeah, well, I think when we think maybe even beyond cancer, I think one of the analogies that people often use when thinking about the microbiome is describing it sort of like a rainforest, like a diverse ecology. And so if you think about, diversity and at that scale around the world, there are a lot of different creatures and a lot of different species in different habitats, but they all have, if there are common functions, that creatures play in different spaces. And so when I think about, when we're trying to learn about, what makes up, a bacterial composition that you may want to transfer into a patient to achieve some sort of effect, you want to start to understand what are the functions that those bacteria are playing in the gut, and how can we select for bacteria that will perform those same functions in a person. And so whether it has to be super personalized and individualized to the particular person that you're working with, or whether there are, classes and types of functions that we just want to be able to restore, I think is an open question, but yeah.
Yeah, I would echo that. I think, will we have a one-size-fits-all, fecal transplant in the near future? Probably not. I think, it will take some time. I think, is there an ideal microbiome that's associated with great immunity, preventing cancer, enhancing vaccine responses and other things like that. Ultimately, maybe we'll get there, but I think it's definitely going to take some time. And I think there's also not only the gut microbes that we can modify, but what we feed them, so diet, nutrition, things like that factor into it. And then all the other things that impact them, like medications that we're taking and antibiotics that could potentially disrupt the microbiome.
Right, any questions from the floor?
Hello, I'm Arthur Brodsky from the Cancer Research Institute. You both mentioned the negative impact that antibiotics can have by wiping out the good balance of bacteria within us. And some that may lead, or I guess it would be common sense almost to think, or be intuitive to think, that probiotics might have the opposite effect and kind of help us out. But I know from some of your recent talks, Dr. Wargo, that that might not be the case, and I was hoping maybe you could share some of those insights with the audience.
Yeah, definitely. And so a very close friend of mine and collaborator, Laurent Sidvogel, showed that if patients took antibiotics just before or just after starting immunotherapy for cancer, they actually did worse. And it was because it disrupted in theory, it disrupted their microbiome and made them less able to mount an immune response. Now, as I think a lot of people have wondered, well, gosh, could I change my microbiome and make it better? And there's certainly off-the-shelf probiotics that sound appealing to patients. And I can tell you, when we asked our patients if they were taking probiotics, 42% of our patients reported taking probiotics. Now, we looked at those patients and characterized their microbiome. And what we found is that if they took probiotics, they actually had a less diverse microbiome, and they actually had worse responses to therapy. Now, this has to be validated, but what I think it suggests, if you think about it, what's a good microbiome? Well, at least we know diversity matters, and I think a diverse microbiome is good. Now, if you take simple off-the-shelf probiotics, often they have 1 or 2 bacterial strains, and if you take 10 to the 11th colony-forming units of that and put it into your gut, you're going to dilute out a potentially good microbiome and make it worse. So I'm not saying all probiotics are bad, but I am saying that we should really exercise caution and really, we need to study these before patients are using them. And I, frankly, would caution against them in cancer patients on immunotherapy.
More questions? Yep.
Hi, my name is Tisha Jepson. I'm CEO of True Bearing Diagnostics. I have a question just related to, my grandfather is a geneticist and went to China to help with the Green Revolution. And what he learned when he was in China was that a Caucasian, could eat a certain amount of rice adjusted for his weight in comparison to an Asian person, and would actually die of malnutrition. Whereas the Asian person eating the same amount would, adjusted for weight and everything, would actually be able to extricate the nutrients. And so do you think that, you were talking about the fecal replacement, do you think that in terms of the metabolites or ways of absorbing nutrition, sort of on the opposite to disease, but rather to health, in the other direction, would be able to be adjustable, since it seemed to be across the board?
Any thoughts on the interaction between diet and...
Definitely. I mean, I think, do you want to take a stab first, and then I can
Sure.
let you know what we've seen?
Well, I think I'm full of analogies today, I guess. I think the one that I like when thinking about, why the microbiome, why on earth do we let these bugs just kind of live inside of us? It's a good question, and I think one answer that I've heard is, if you think about it, the human genome like hardware, these bacteria are a little bit like software, allowing us to evolve, as humans spread out around the world and we're exposed to new diets, new food sources. The bacteria that they picked up in their large intestine ended up, being pretty critical for being able to break down and absorb nutrients from their food. And so, over the course of human evolution, I guess, that story and that relationship between bacteria in our guts and our ability to absorb nutrients from our food is, I think, well told. But if you want to speak more.
Yeah, I think I can speak in, certainly there have been, it's provocative to think, could we actually change the microbiome and help people digest food better, and, eliminate obesity and, all these things. That we're way off from that. But what I can tell you is that we have looked at diet in our patients who have cancer who are going on to immunotherapy, and we've seen if they eat a better diet, i.e. high fiber, they actually have a more diverse microbiome, more of the favorable bacteria, and better responses to treatment. Again, we need to study this further. We need to look into what aspects of those diet really is potentially driving that. But I think, can we enable our patients to really to enhance their own responses by eating the right things? We just need good guidelines, but we're working on it.
I think it's fair to say that for the moment, the science of the microbiome is very much descriptive. We're looking at differences between different populations. And the next step for the field is to get to a point where it's firstly predictive, where you can actually look at someone's microbiome and work out stuff about their biology that's relevant to their health. And then a thing you can actually manipulate, where we can, with precisely and effectively, change the microbiome in a way that we can improve people's health. And I think we're not quite there yet, but there's definitely a lot of interesting science that's going to take us in that direction.
Yeah.
But thank you very much. I think that's all we have time for. Thank you both so much for joining us.
Ed.